All right. All right, so today we're going to move on and we're going to look at how we measure the evolution of a population. So we talked about population evolution last time, and now we're going to look at how exactly do we measure it? Or, wow, can we measure how much a population is evolving? And the idea is we can, relatively speaking, and we can actually with a um, quantitative method. And so that's what we're going to be looking at today. It goes along with something on your AP biology formula sheet called the Hardy-Weinberg equation. So let's not forget about what we talked about last time, the agents of evolutionary change, right? The five mechanisms uh, that propel change by evolution, which is mutation and gene flow, non-random mating, genetic drift, and selection. So we had talked about these as really the core principle from last time about evolution of a population, and we just want to remind ourselves of these factors as we start today when we talk about measuring evolution of population. So conceptually, a population, like we talked about last time, is a group of organisms of the same species that interbreed, that reproduce with each other. And we say the gene pool is just a collection of all the alleles in that population. So remember, alleles are the different copies of the gene. So for example, if we were looking at the gene pool for a specific trait, remember each organism would have two alleles, and think of those as marbles. Maybe a red marble represents a dominant allele and a blue marble represents a recessive allele. And if each individual in the population had two marbles representing their two copies, and we put all of those marbles collectively in a bowl to represent all of the alleles for that gene in a population, that would be the gene pool. Now, the allele frequency is how common a specific allele is in that population. So if you think about it probability-wise, if each of those, if a red marble represented a dominant allele and a blue marble represented a recessive allele for a specific gene, and we put them all into a bowl representing the gene pool, the allele frequency, all right, would be how common the red marble is in the bowl or how common the blue marble is in the bowl. Evolution at its most basic definition, is a change in the allele frequency in a population over time. So it doesn't mean in a specific direction, just that the frequency of the marbles in that dish is going to change as time goes on. So we want to kind of think about what conditions would cause allele frequencies not to change. So as generations go on, what would cause those frequencies not to change? So a non-evolving population, we need to remove all of those agents of evolutionary change we talked about last time and we prefaced this time with. So we would need a really, really large population size so that genetic drift, those random chance events, don't occur. We would need no gene flow, so no organisms are moving in or out of our population. There's no variation, so no mutation happens to change the gene pool at all. And that there's random mating, so we get rid of sexual selection. So just kind of random, no no way to figure out who's more likely to be sexually selected than the other. And then most importantly, there's no natural selection. So each organism, independent of their assortment of genes, is equally fit to survive and reproduce. These are what is necessary for a non-evolving population. Okay. So what we're going to do to be able to measure the evolution of a population in a quantitative way is we're going to compare an actual population and the allele frequencies in an actual population over time to what the allele frequencies would be in a non-evolving population. And the greater the difference, the more quickly the evolution is taking place. So this brings about the idea of what's called Hardy-Weinberg equilibrium. And it is a hypothetical, it's a non-evolving population, and it is something that we can compare actual data to in a relative way to figure out how fast evolution is occurring. So in a Hardy-Weinberg equilibrium, allele frequencies always remain the same, since we said that evolution is a change in allele frequencies over time. So it serves as our model to compare against. And there is no natural population that is in Hardy-Weinberg. I shouldn't say rarely. It's not possible for a natural population to be in Hardy-Weinberg equilibrium for any discernible amount of time. So it's useful as a comparison, as a null hypothesis, to measure the forces acting on a population and to measure the relative amount of evolutionary change. So this is the Hardy-Weinberg theorem, or the Hardy-Weinberg equation, and it has to do with counting alleles. So assume that there's two alleles for a given trait, capital B, dominant B, and recessive B. We are going to represent the frequency of the dominant allele in the population as lowercase p. Okay, the frequency of the dominant allele is lowercase p, and the frequency of the recessive allele is lowercase q. So when we talk about allele frequency, that's equal to number of specific alleles divided by number of total alleles. So to find the frequency of a given allele, you just take the number of, the dominant allele or the number of recessive alleles in the population and divide it by the total number of alleles. That's the frequency. So because it's a proportion, they have to add up to 100. So P plus Q needs to be 100, or P plus Q equals 1. So the Hardy-Weinberg equations are a group of two equations, and this is the first one. P plus Q equals 1. Let's look at these cats right here. All right, there are 1, 2, 3, 4, 5, 6 total alleles in this population. 1, 2, 3 of them are dominant. So 3 over 6, or 0.5, would be the frequency of the dominant allele, and 1, 2, 3 over 6, or 0.5, would be the frequency of the recessive allele. So 0.5 plus 0.5 is equal 1. Okay. Hopefully we can convince ourselves of that with that simple demonstration. The frequency of the homozygous dominant phenotype or genotype then is the frequency of the dominant allele p squared, p times p, or p squared. The frequency of the homozygous recessive genotype, having two recessive alleles, is q times q, or q squared. And the frequency of the heterozygote is p times q plus q times p, either way, associative, all right, or commutative, sorry. Either way, you get the same thing, 2pq. So frequency of all individuals must add up to 1. So the second Hardy-Weinberg equation is p squared plus 2pq plus q squared equals 1. All right. Again, frequency of the homozygous dominant phenotype, p squared. All right. Frequency of the homozygous recessive, q squared. Heterozygote, 2pq. So the formula is in terms of alleles and in terms of individual genotypes. These are the two Hardy-Weinberg equations. And basically what they're saying is that if a population is in Hardy-Weinberg equilibrium, these will be true for each generation. So the frequency of each allele won't change from population to population. So we can use these to calculate the individuals and alleles of the next generation, if a population is in Hardy-Weinberg equilibrium. So let's say, what about this? There's a population of 100 cats, 84 are black and 16 are white. How many are of each genotype? So the frequency of the heterozygous genotype, well, 16 white divided by 100 total is 0.16. And that means the frequency of the recessive allele is just the square root of q squared. Square root of 16 is 0.4. And then we can figure out the frequency of the dominant allele by saying, okay, p plus q needs to equal 1, so 1 minus 0.4 is 0.6. That's an example of how we could use this equation. Just a reminder, we always have to know or make the assumption that the population is in Hardy-Weinberg equilibrium to use the equation. Right. So let's say that we were starting out with this. We have the genotypic frequencies, assuming Hardy-Weinberg equilibrium. That's kind of our default, our null hypothesis. We would assume this. And then if we get in the sample data, look how different it is. How would we explain these differences? Well, these differences between what we would assume we should get if the population was in Hardy-Weinberg equilibrium and what we did get show evidence that change, that evolution is taking place. And the greater the difference between these assumed hypothetical numbers and these sample data, the more quickly and strongly selection is taking place. So let's do another example. Let's see if we can figure out allele frequencies for a population of pigs using this picture. The goal is to find q squared, pq, and 2pq. So can we identify who's the dominant and recessive, right? So white or pink coats are dominant over black. Can you determine who has what alleles? Well, only really for the homozygous recessives. We know the black pigs have to have 2 recessive alleles, but we don't know if the pink ones are homozygous or heterozygous. So we're going to start with the black ones. Let's count how many there are. 1, 2, 3, 4 black pigs out of a total of 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 in total. So what we're going to do is calculate q squared by counting the individuals that are homozygous recessive, the black ones, and then calculating the percent of total they represent, because that will be our q squared, the frequency of the homozygous recessive genotype. So we have 1, 2, 3, 4 out of 16 is 25%. So q squared is 25%. And then to find q, we just take the square root of 0.25. This is 0.5. So q is 0.5. The frequency of the recessive allele is 0.5. And we know that p plus q must add up to be 100. So if we subtract, we can find that the frequency of p is also 0.5. To find pq, well, that's pretty easy because now we know p and q. So we just multiply 2 times 0.5 times 0.5, and we get 0.5. So this shows that the allele frequency in generation 1, okay, 0.5p, 0.5q. One would study the allele frequencies on several generations. We look at their offspring to see if the population is evolving. If it is in Hardy-Weinberg equilibrium, we would get 0.5 for the allele frequencies in each successive generation. So what's the point? Why do you use Hardy-Weinberg equations to determine if a population is evolving? Well, when we calculate the equilibrium in the beginning of a population, we know the distribution of alleles. If we examine a population later in time and we see that there's been a change, then it's evidence that evolution is taking place. The greater the change, the more quickly evolution is happening and the more strong selection is for that specific trait. Okay. That's kind of where I wanted to end here today. We'll go over much more of this here in class together.